Great. So let me pull up slides. So yes, good afternoon, everyone. I'll walk you through over the next about 45, 50 minutes, some of the applications and intrigue actually for epigenomics, which I'm sure most of you have heard about or have been thinking about, maybe even already been working on some of this, but I'll walk you through some of the applications for it and some things that we've learned, in the past few years. Let me share my screen. Let me put this up here. Okay, great. Can everyone see my screen? All right. And like hide the floating meeting controls. All right. So this is all about epigenomes, DNA modifications, and also some epitranscriptomes. So in the first lecture and in some of the previous lectures, if you all recall, we talked a lot about precision medicine, and there's a lot of work, of course, at Cornell and at many institutions around the world to go from anecdote-based medicine, I would say, which is where we're looking at groups of people and vague memories of what a clinician may have seen, into a molecular-based guidance for, say, cancer or infectious disease or care. And so this is the bedrock of a lot of what you're learning in this class and other classes, and most of you, there's graduate students, medical students, or researchers. This is the world in which we live, but we take it for granted. It took a while to get to precision medicine. But what I want to think about today, and I think most of you know this already, is to think about not just precision medicine, but also think about predictive medicine. And you can see here, we launched an initiative a few years ago for quantitative prediction. And here we look really, like we're looking at this mysterious laptop here, but really it was a screensaver. It was not as exciting as you might think. We look really enthralled, but it's just a screensaver. But we launched this large initiative, actually, at this point, now almost 8 years ago, was to think, how do we build more algorithms into predictive medicine? So not just getting a good diagnosis, matching it to the right therapy, which of course is essential, but how do we start to think more long-term about predictive medicine and ways to do tracking genetics, epigenetics, how do we modify some of the epigenetics? So we'll talk about that a bit today and ways in which we can do so. So the model really of what I think of a lot for medicine, of course, you want to, if you are sick and you do get a disease, whether it's a cancer, a neuropsychiatric disorder, really any disease that might unfortunately erupt in your life, you want to get precise diagnosis. But could we find it before it occurs? And I'll talk about this a little bit today and also in a couple of the later lectures about ways to predict maybe even 5 or 10 years in advance before you get sick. Are there ways to find it and even prevent it? And I'll talk a little bit about that today as well. But this idea of them continually informing each other is something that we can and do think about really all the time. This has led to the All of Us program, which is also on campus. If any of you have not done yet, you can get your genome sequence for free at Cornell and register and get your blood drawn. Due to some of there's a bit of dark humor in the lab this past week, is that because of the budget cuts, maybe it's no longer the All of Us program. It's like the Some of Us program at the NIH because of we're losing money. Or they closed down their lab. It's right next to our lab, and they closed down their blood collection, which is a little sad. It's like, oh, it's the Some of Us program. But this is being recorded, so I'll just be politically careful, but say it is a really great resource. And we want the money to be, returned, I guess, to what was the ambition, was to do 1 million people and have their genome sequence plus their clinical metadata and use that to build better models, which is similar to, like, say, the Million Veterans Program, which also is building molecular databases plus also some of the, phenotypic data, the medical data. So this helps us give better care for the Veterans Program, for example. It's a really large-scale project in the United States. And as I mentioned a bit, as of last year, there's a lot of projects in almost every major country has some degree of a population genetics program. Here's a short-term list of most of the ones I know of as of last year. But this is now fairly standard. Many large-scale, tens or hundreds of thousands or even millions of genomes being sequenced at one time. And that's at the genetic level. We talked about that a lot in the first lecture, but I want to think about both genetically and epigenetically what changes as we age. So, of course, we think about the genome as the bedrock of some of the diagnostics and, of course, predictive metrics for your biology and risk of disease. But the genome is actually not totally stable. Of course, as we age, one of the things that changes is telomeres, which are the caps at the end of your chromosomes. They shrink over time. So your average telomere length gets a little bit lower, and I'll talk about this a little bit later with one of the papers. And eventually, when it gets too short, the cells will basically apoptose and die. But this is one thing that changes as we age, telomeres. And this is something I was alluding to, is that, other things that occurs if you look at the frequency of people that carry mutations in genes that are called clonal hematopoiesis genes, which means the genes in your bone marrow, you're constantly making new bone marrow cells and actually hematopoiesis to make all the cells that create blood in your body. but they accumulate mutations as all cells do. And actually, if you look at what percentage of your cells are carrying these mutations, if you live long enough, eventually you will carry some of these mutations. But what's really fascinating is a study that was led out of Weill Cornell showed that you could maybe predict as early as even 18 years or maybe 10 years, depending on the patient, that before someone gets leukemia, that you can track their mutational trajectory that they seem to be at high risk. And so this is something that's very interesting as a predictive mechanism. Again, predictive medicine. Could we see that you're on the way towards leukemia and do more active monitoring, maybe even do some prevention before you would get there? So this is kind of another fascinating concept of that. Genetically, you think, oh, I've sequenced my genome here in my 20s or 30s or 40s, but it will change as you age. The number and type of mutations that you carry will increase, and all of you are more mutated today than you were yesterday, but you all look great. Don't worry, you look great, but you're definitely more mutated today than you were yesterday. But this brings up, so that's the DNA which changes, but there's also the epigenome that changes, the regulatory landscape behind and around DNA is how DNA is packaged and methylated and compacted. And I'll walk you through a heavy focus today on DNA methylation because a lot of it links to some of the sequencing methods. But we'll talk about this regulatory layer behind DNA. So in particular, there, of course, is the 4-base pair genetic code, which you can see, mostly in black. But the epigenome marks are, there's either how DNA is packaged or modified, refers to often the epigenome, where it's the part of the genome where the ACGT doesn't change, but the regulation of it and the modification of it does change. In this case, there's 5-methylcytosine, hydroxymethylcytosine. There are many chemical modifications. There's formol carboxylcytosine, 6-methyladenine. I'll talk later about 8-oxoguanosine. You can see in adenosine here. These are damage bases that appear when you have radiation or reactive oxygen species. So these are some interesting, base battles that your DNA has to endure, and they're manifested in some cases down here. But other ones are just these chemical modifications that are slight tweaks to regulate your DNA, and I'll come back to that. So one of my favorite papers I've ever actually read. I was an undergrad and was reading this paper because you can just tell how old I am. This was from the year 2000, but it was about an egg and sperm right when they come together. This is from a mouse embryo, and I don't know if you remember your embryogenesis. I don't know if you remember that moment when you started as an embryo. What I remember is that there was Marvin Gaye and there was some candlelight and there was I think really soft lighting. That's what I remember. I don't really actually remember, but that's what I imagine I should say. I've asked my parents, they refused to give me any information about what exactly happened at that time, except I know that I was unplanned. It just goes to show you what plans give you. But in any case, when egg and sperm come together, whatever you imagine your embryogenesis looks like, this is in that moment, literally hours after egg and sperm come together. And this, again, it's a mouse embryo, but the human embryo is very similar. Is that you can see egg and sperm are together. There's two haploid genomes. They've come together. There's a eureka moment. But then the paternal genome you see is demethylated quickly. All these green marks indicate the antibody for methylcytosine. So you can see the paternal genome gets rapidly demethylated. Those marks start to disappear. The green goes away, meaning that as soon as fertilization happens, the paternal genome starts to get reset. All the epigenetic marks get dropped down basically to zero. The maternal marks also eventually go mostly to zero, but after more through division, it starts to get almost basically diluted. And most of the epigenetic marks in the genome, the methylcytosine marks that can help control gene regulation, all get dropped back down to zero. If you look at a paper that showed this with great detail from 2012, so that was a very early paper in 2000. This got detailed at fine resolution in 2012 to see, okay, egg and sperm, you can see they're, rapidly all demethylated. There's less in the oocyte, but then this is the number of cells as an embryo. Once you look at the inner cell mass, it means that in the early days of the embryo, you can become any cell in your whole body, which is one of the it's literally the reason I became a geneticist because of this plot. Well, I didn't see it at the time, I was too young, but the concept that you can have one cell that has the potential to be all the cells in your body and the instructions are just there, I still was, and I think is one of the coolest things I've ever learned. But what's interesting is once the inner cell mass reaches this state of pluripotency, or really totipotency, because it can become all cells in the body, then you have to start to pick. You have to say, I'm going to start to make other types of cells. I'm going to make other cells become part of the trophectoderm. You're going to make different layers of your body that become different tissues of your body. And this is paternal and maternal, and some of them don't change that much. But you can see, let me jump back to this. This is what changes. You start to make all the tissues, and you go through, basically become a fetus, and you eventually are born. But you have to make all the cells in your body from this very first state. And the way that you do that is to both create some of these methyl marks and actually sustain them is how you start to pick which lineage you've chosen. What cell types are you? And I'll come back to this in a little bit, but this is one of the, I think, most beautiful follow-up studies from an earlier paper that shows this quite clearly. And the other interesting thing about this, if you think about identical twins, because they're both embryos that were split in two, and then they had the same reprogramming that happened, and they differentiated. When you look at identical twins that are 3 years old, this is the number of differentiated methylated bands. You can see they're pretty similar. But by the time they're 50, they have more epigenetic changes between them than they did when they were younger. These are not the exact same twins, they're just donors. Because it's very simple. The number of bands is greater. That means they have more epigenetic changes. They also, as you just heard from the previous slide, they have more genetic differences because they're all accumulating mutations, which is, of course, inevitable through just living. But you can see, this difference. That means that you're genetically and epigenetically changing as you age. Some of it's because you want to differentiate into cells like neurons and cardiomyocytes and other cell types. But some of it's just because we age and these methylation marks do change over time. And so because of that paper in 2005, actually, there's some great work by Hannon in 2012 and Steve Horvath in 2013, that he just had that instinct and said, well, if we just take all publicly available data that has age and DNA methylation, put them together and build a classifier to say, okay, here's all of our training data, here's my test data. Can I predict how old you are based on DNA methylation alone? The answer is, it's a little bit noisy, but this was the model, and the answer was like pretty well, actually. This epigenetic age increases in all tissues as time passes. These are the different tissues that we're taking from public data. So it's pretty consistent. It's a linear trend. Again, it's noisy. It's noisier in some tissues than others, but it's definitely linear and it increases over time. And this was validated by other groups the next year. So this is their predicted age versus chronological age. So this epigenetic age, which is just methylation based, you can see it increases over time, and it's, plus or minus a few years. It's not going to tell you what month you were born, but it will increase over time. And one of my favorite aspects of this paper is they took induced pluripotent stem cells and embryonic stem cells and looked at their predicted age. And you can see here, like that paper I showed you from 25 years ago now, their epigenetic age gets down to zero. Actually, it's minus age because of the linear model. But you can see they reset all the marks back to zero as expected based on that earlier methylation data. So this is a fascinating feature of how DNA methylation works. And so this opens up some questions. Like people are starting to, of course, create companies about this and how old are you? There's a number of different consumer tests that are out on the market that will try and tell you your epigenetic age. I just took one last week. I'll let you know, hopefully within a couple of weeks, what my epigenetic age is for the class. We can all take a poll, maybe. Probably wide range. We'll see. But what's interesting about that is how old are you is on the methylation markers, but we're still figuring out new tools in the molecular toolbox. And what I mean by that is historically we've known of a number of genes that create these methylation marks or demethylation marks here. I'll come back to this in a second, but some of them were just found a couple years ago. These are new enzymes that control or modify methylation just as of a few years ago. So we'll put all these papers up on the course website, but we're still finding these tools. That's in health and in normal aging. But what about in disease? This is the other big point, is that methylation, an older model, again, this is from a paper about 20 years ago, is the idea that you have these methylation marks. These kind of look like lollipops. They're actually called lollipop plots because of that. So black just means methylated, and white, these open circles here, means unmethylated. But at some point, you do need some methylation, as I was just showing you, in your body at different places. But if you get too much methylation at one spot, that might actually turn off genes or disrupt genes, and that could lead to more of this has been seen in cancer. The original paper on this was actually by Andy Feinberg, at this point almost 40 years ago, showing some differences between methylation and cancer. But this is basically the model. And what's interesting about this is, if you look at cancers like leukemia, for example, what are the genes that show the most mutations? Many of them, these arrows show here, are epigenetic modifiers. So you think, I'm getting a mutation in a gene that could be bad, but that gene could be an epigenetic reprogramming gene, which then itself leads to the cancer. So we often think of cancer as both a genetic and epigenetic disease. I'll come back to a very interesting example of that in a second. But this is very true in leukemia. And this is, what we've seen for other patients, that if you have mutations in these genes, IDH1 or 2 or TET2, there's impaired DNA methylation processing. So this leads to increased DNA methylation around the cell. So instead of, this wild type gene processing alpha ketoglutarate and stopping, you get mutant IDH1s that makes what's called an oncometabolite called 2-hydroxyglutarate that prevents the proper functioning of this cycle of going from methyl to hydroxymethyl and back again to regular cytosine. So once you lose the ability to do appropriate transition between methylation states, it eventually can lead to cancer. And indeed, if you look at those, this is a Kaplan-Meier plot. If you look at the survival curve of how many patients survive, if you have these kind of mutations, these patients do much worse than the patients who don't have those mutations. So this is a very poor prognostic marker if you have these epigenetic mutation factors. Now we've published a range of tools and methods called one's called MethylKit or EDMR over the years, different tools. Sometimes like I used to say that our lab is full of meth addicts, but now I stopped saying that because that led to people getting the wrong idea. But we like methylation, at least I should say. And if you look for meth lab in image search, you'll find all sorts of things and that you probably shouldn't have in a lab. But in any case, methylation in this context, meaning epigenetics and tools for mapping and programming these modifications, which we now call EpiSuite. So there's been a wide range of different tools, and some of which you'll be learning for the course is how do you process single molecule data to look at base modifications, which you'll learn about. Might not be next week, but in I think 3 weeks will be some of the base calling modification homework. So I want to go back to this chart again. We talked about where you see these methylation marks, and so each one of these little black marks here you can see is a CPG island, and you have genes here in gray, and then you see a CPG island. What that means is if you have actually I don't have it right there, but the CPG just means you have a cytosine, a phosphate, and a guanosine along a track, and if you suddenly see a lot of CGs together on 5' strand of DNA in a big cluster, this island is right in front of the gene. And what's extraordinary is that right in front of most genes across your genome is this little dip in methylation. This is exactly where the methylation drops down to near zero, but then it comes back up again. And in normal tissues, it stays pretty regulated. But once you have cancerous tissues, you start to have this drift. You lose that regulation, like I was alluding to before, of this methylation machinery, and you start to have places that are normally hypermethylated become hypomethylated. You start to see this drop here. And so instead of being at 0.8, it goes down to 0.2. You lose this regulatory control of your genes, and genes that are supposed to be turned off suddenly get turned on, or genes that are on get turned off. And so this is, of course, bad for maintaining cell states. And so that's what we've looked at in broadly across cancer patients. But one vignette I want to show you here quickly today is there's one patient we looked at we called Chernobyl Man who had unfortunately cleaned the nuclear waste site after the explosion in 1986 and then sadly developed leukemia. But we had a unique opportunity because his samples had been banked over the course of almost 2 years. And so we could see the differences, say, genetically or epigenetically in this patient where we had relapsed multiple times or had been refractory to therapy again multiple times. So very fascinating cancer patient. It's also interesting. Most cancer patients, if their cancer comes back, they will go to a different doctor. It says a lot of times, but not always. But this idea of having the tenacity to stick with it and also collect samples was unusual. But this is what was interesting is there's both genetic evolution on the right and epigenetic evolution of this cancer. We could see how many sites are changing across the genome of these epiallele sites per million reads would jump up at the first remission, first relapse, and whereas somatic mutation, the number of new mutations was relatively flat and then came up more later. So we can see there's differences here between genetic evolution and epigenetic evolution, at least in this patient's leukemia. And we could also see areas where is it the same locus? Here we could see genetically on the right, most of the mutations stay the same throughout the cancer. They get some new mutations every time point, but the epigenetic dynamics were much more dramatic and much more significant. We saw a lot more changes at every stage of the tumor's evolution at the epigenetic level. And so that was also interesting to see. If we plot this a different way, you can just see this like low frequency variants here that were popping up, particularly here in the later time points, which is partially part of the difference of what may have been driving the final stage of the cancer, unfortunately. But this low epigenetic evolution we think was antecedent to this low variant of evolution at the genetic level, which is again kind of an interesting. These two different tracks and two different trajectories for evolution of this cancer, both genetic and epigenetic. And so how we did this, just to give you some background, is we would take from a single read. If you have 4 sites, it's basically they're binary, so it's 2 to the 4th power of possible combinations. You can see here kind of all the combinations. If you have 4 CPGs in one fragment of DNA, and then we built this into an algorithm that would then track out this entropy. So actually, the original formula for entropy was from Boltzmann, which is the number of states in a given macro state. So really just taking some old thermodynamics equations to see how much entropy do we have. It has also been applied to amino acid sequences and also the mutation assessments. But we took that same formula and applied it to the amount of entropy per site and the number of epilleles per million loci. And that's how we got this calculation. So when you start to get, because you'll look at this for your homework, is when you get single molecules of DNA and you can see individual patterns that are phased, and phased means they're on the same molecule, you can see multiple changes. You can start to create these interesting patterns of How much is evolution changing at one locus, and what proportion of reads are carrying consistent or divergent epigenetic differences? And so this is one example, at least from our group, that showed this difference. And so we took that metric of EPM at diagnosis versus relapse, or relapse versus the normal, or relapse versus diagnosis, to see what's the degree of change. And what's notable about this is that in some cases, this is within the same patient, relapse versus diagnosis, over a course of either a few months or maybe a year and a half. In some cases, there's millions of sites that have changed. In some cases, there are 0 detectable sites that have changed epigenetically. So sometimes it's a really wide range, everything from nothing observable different to massively, millions of tens of thousands of sites changed. And this is what was so fascinating, is that it really, if you think of anyone who's ever had a cancer, you always have to think, well, was it a genetic reason or an epigenetic reason? And sometimes it can be one or the other, from what we've seen. So this is something we published here, but then we also want to say, well, okay, so if you have progression, does this change your phenotypic outcome? Do you have a higher or lower, worse outcome and a risk factor? And you can see those patients with more epigenetic dynamics and have a faster evolution had a worse prognosis. So it has some indications for what you could look at for your prognostic relevance as well. So that's what we published this a few years ago, and this was showing the different evolution and dynamics of both genetics and epigenetics in AML, and kind of separate paths for genome changes in cancer evolution. But I want to point. So we published this, and other people have published related work, like this one, for example, that shows that these epi alleles can look at high risk versus low risk patients. This was kind of a follow-up paper to some of our work that confirmed these results. That just if you look at that heterogeneity, that entropy, like Boltzmann's entropy, you can see differences of high risk and low risk patients. But I bring this up because there are many, there's also cell-free DNA has potentially early detection tests using methylation. So there are other tools to look at methylation to predict your risk of cancer, or cell-free DNA metric. So this is being used by a number of groups, not just ours, or not just at Cornell, of course, but many groups looking at this. But then this I want to transition because it's very disappointing to tell a patient, well, do you have a bad cancer or a very bad cancer? It's you'd rather be able to say, well, we know what you have and we think we could target it. So some of the ways we've looked at targeting over the past few years is, for example, combination therapies. Can you combine different drugs together and then treat them, say with azacitidine, or in this case, AC220 was an experimental drug by Ambit, and another one that's by Agios. In this case, this was from a mouse study. But here we could say, what happens if you do combined drugs? You see plus plus when you do both drugs. And looking at these same methylation differences, you can see if you do both drugs, you can get back towards the regular state of CpG. Instead of hyper or hypomethylated, this shows that you could get back towards, this is the degree of difference you can see on this axis. This shows it's possible to start to bring it back towards a baseline. And here, if you look by color, you want to get back towards basically this green combination, then you're almost back to 0. So it shows that you can, broadly reset epigenetic marks. But that also begs the question, can we detect them better? We can, maybe target them better. And I want to say, well, then can we also target them better? And this gets back to some of the work we talked about in the very first lecture. If you remember the PacBio sequencing, where you can actually look at base modifications here as you look at a polymerase, because the speed of incorporation indicates whether it's a modified base. We already talked about the Oxford Nanopore single molecule sequencer, and again, this will be part of the homework. But and as I mentioned in the first class, also new methods need new standards. So we talked earlier about these genome and Abano standards, and I'll review this quickly because some of this in the first lecture. But you can do this anywhere on Earth, but also in space. And we don't want to just hold meetings. Remember this, as we don't just have meetings to talk about standards. We want to actually make sure we can use these for specific benchmarking. This is the GM and Abano consortium, which I mentioned in the first lecture, but I want to just highlight here. These same standards were used for the long read platform that you'll be looking at. It started way back in 2014. It was because we wanted to look at RNA sequencing, but now actually this is across all platforms, which then was the FDA SEQC study, the EpiQC study. So these are the studies to look at differences of each of the kind of sequencers. We like to say we were bringing sexy back, but nobody says that but me, but that's what we're saying because it was sexy part 2. But this genome in a bottle standard was a way to actually have a well-characterized and uniform human cell line that we could use to characterize how well we can detect epigenetics, and also used it for other diagnostics. Again, there's GitHub repositories for all these tools, which I mentioned before as well. And these samples are the ones we use for the genome in a bottle samples, and also the EpiQC study, which is relevant more for today because we're talking a lot about epigenetics. So I mentioned this briefly in the first lecture because we talked about these different platforms. So this is a bit of a review. but these are the 4 genomes that were looked at, and they're trios. And there's also the methylation data that was done across all these different platforms. So now that you've heard about methylcytosine, you can have a better appreciation of 5-methylcytosine detection. Here, when you do a base conversion, if you do a bisulfite treatment, the methylated cytosine you can see here stays the same, and it still shows up as C after you do PCR and sequencing. If it's unmethylated, it gets deaminated, shows up as a U, and then becomes a T after PCR. So you can count per base where you see basically the deamination event or not, and there are a range of different methods you can see on the right here for actually doing this kind of bisulfite treatment and then ligation and sequencing. There's also enzymatic methods of deamination using ApoBec enzymes and TET enzymes. So you can see here there's another method from NEB, for example. And we used all these methods and compared that also to Megalodon at the time was the old base caller. You'll be using Dorado for this course, but this was the old one at the time. And we did a comparison across all these different platforms for how many reads get mapped, how many of them are duplicated, how many of them are unmapped. What do we see for the different accuracy across all these methods? And I bring this up because this was the first chance, about 3 years ago, we got to actually look at a really robust examination of nanopore sequencing for methylation versus the more established methods that have been around in some cases for 20 years. And this just gives you a lot of statistics from the paper. But then we did downsampling and compared across all these different platforms. This is just some of the technical details from the paper of how we get them to be comparable. And here's what was interesting: is if you take a look across these different methods, you can see that, as I showed you before, there's that thumbprint of evolution is at the transcriptional start site. You see this dip in methylation, and that's expected. That's exactly what you'd expect to see. It's consistent across all platforms. But if you look at the overall coverage, remember, look at all the reads mapping at one spot. It was a little bit noisier for some platforms than others, which wasn't really totally expected, but it was interesting to see that show up. But we did see this dip consistently as a percent of methylation, even the coverage was a bit variable. But here's where it got really, I think, interesting, and it's when Nanopore, at least to me, really became very mature. Is that suddenly we looked at the correlation matrix of Nanopore versus all these other methods for calling methylcytosine, and we could see here that it was actually, R squared, R of 0.9, 0.99, 0.91, 0.92. It's pretty good. It's not as good as some of the established methods that have been here for decades, but it was, 0.01 or 0.02 difference in some cases. So very correlated overall across low and high methylation. So this is where I think we really started to get very excited about the nanopore sequencing because you could get whole genome sequence data and methylcytosine at the same time, and it was starting to actually be really comparable to existing methods. And even at 20x coverage, because if you go back here, you can see you get some variability in coverage. Then we downsampled this coverage to say 20x, still look pretty good. Even at 10x, the lower coverage of the genome, less molecules per region mapped, we still had really good quality mapping. You can see here for the coverage and correlation. So this just was really an exciting indicator that it was getting ready for prime time as a methylation platform. Because I can't resist, say, well, okay, does that happen in space? Does this matter if you're in space? So talked about this briefly before, but we had the NASA twin study where Scott Kelly went up in space for a year. And now we also have Senator Mark Kelly who's here on Earth and look at the differences during the twin study to see are there epigenetic differences along a host of other differences. So we did look at methylation, RNA methylation, metabolomics, many other omics for that study, as you can see here in this slide, to make a multi-omics analysis of human spaceflight. And one of the things that we saw was indeed many things changed. Some things, a lot of microbiome species, proteins, cytokines increased. Telomeres got a little bit longer in space, which is the opposite of what we expected. I'll come back to that in a second. But the other thing from this study is that we could see, if you look at purified CD4 cells or CD8 cells, if you look at the overall noise, epigenetic noise here, you can see of methylation cytosine, there was a little bit of difference, but mostly consistent. But if you look at specific pathways of which genes are related to different pathways, this is where we saw some of the bigger differences in T cell differentiation or, like ossification, looking at bones. That was kind of fascinating to see. So we could see some differences in these immune cells related to where we're seeing most of the methylation changes across the genome, which was, intriguing to see. And we published this a few years ago, so I'll mention this briefly. But that was one of the changes. Since then, we've been looking at, how many things do we see of someone who's gone to space versus someone on Earth. And we could see it's actually, if you look at the overall methylation difference of someone on Earth versus in space, there's actually a lot of changes that just might happen over time because of epigenetic age and diet and stress on the bio. So ironically, even though there were these changes within one astronaut, they were not as dramatic as some of the changes we saw from someone who's just living on Earth and having pizza and margaritas for a while. So we did calculate the epigenetic age and biological age actually for both twins, and you can actually see here They were, mostly stable. You can see the chronological age in red kept increasing, but it's pretty noisy, as you can tell from those previous plots. You can get someone's approximate epigenetic age because you can see they're here about 50, 51, 52, and all the metrics are between, 47 and 53. So you're getting an approximate age, but it's too noisy to say, did they age 12 months versus 6 months in space? But we can see it is possible to get their epigenetic age from these metrics. To jump back to the telomere question quickly, because speaking of age and genetic age, we did look at telomere elongation in two other astronauts and confirmed that for this mission. And we also looked at this for people that had climbed Mount Everest. There were some twins here. Matt Moniz, who was climbing, and his identical twin, was also climbing with him there, another colleague, and then he had a twin who stayed at sea level. And so we compared those two on mountain twins, also got longer telomeres, and these is 11 other astronauts from this study. So this seems to be a fairly consistent feature of spaceflight at this point. But I want to use that as an exemplar to talk about the long reads for about another 5-10 minutes here. What's the power of the long reads and what else you can see when you have these long reads? So one of the first things we looked at is we got this qPCR result from the twin study, but we wanted to actually say, let's take long reads and get a single molecule map of telomere length. Instead of just getting qPCR data, we could actually see here that at first it didn't work that well. If you look for TTA GGG, this is the canonical telomeric repeat. It didn't always map. This is the mapping quality, and you can see it was really just not mapping. Couldn't see anything. But then we also started to look at the base calls. If you'll have this experience a little bit in the course, is if you change the version of the software that you use, the results can sometimes change. This actually, to me, is one of the most terrifying plots I've ever seen. This is from Kirill Gregorov, who was in lab, and he said, and I was like, well, maybe let's just try different versions of Guppy. How different could it be? And then look, recall the bases from the nanopore data, and then calculate the motifs in the data. And I thought it'd be slight variations, but instead we sometimes would completely lose the ability basically to call TTA GGG, which is the repeat. Again, it's the same raw data, just recalled on which bases are there, which was terrifying just to see this plot. But it got better, it got worse. Anyway, it's calmed down a bit. I know Teo will comment on this probably later and Juan, but like it's mostly calmed down. Anyway, it's a sign of innovation if you're constantly changing things, I guess, but it's a real pain if you're trying to run code on some data and it keeps changing. But this just goes to show that the computational methods for base calling can be wildly important. And those aren't the telomeres we were looking for. We're very upset about those ones. I felt like really stupid stormtroopers. Does anybody catch this reference? Any Star Wars fans in the audience? Okay, I got a few of them there. Okay, great. But we wanted to actually be able to map the telomeres quite accurately. So we wanted to say, okay, can we look at subtelomeric alignments and map them and calculate the lengths? And this was built by Kirill Gregorov and John Fuchs and Daniela in the lab, who said, okay, we're going to map the reads, basically chop out the ends, look for motifs, scan for the appropriate cameras, and then make a density plot of when we've hit the telomere. We should be able to see it because we should see the density and spike up of the expected motifs. And indeed, when we did that cleaning, we could start to see the density map of TTA-GGG. You can see here is in green. It pops up. We can see, aha, we're not, these are, tens of thousands of bases per molecule. Each line's a molecule. And we don't see it, don't see it, don't see it. And suddenly, boom, you can see you're in the telomere. The density of that motif pops up. So I went from one of my least favorite plots to one of my favorite plots I've ever seen. It's like, aha, this is very clear. We've hit the telomere. We're beginning a burst of this density. We did also look at PacBio reads. Actually, that data was a bit more clean. You can see that burst goes up to almost 100% of this density of TTA-GGG, which is the canonical telomere repeat. And you get to the end of the molecule, and that's the end of the chromosome. So we did confirm that the in-flight time points did get longer and published that in one of the prior papers at Luxton, and also published the methods and a lot of more details in this paper a couple of years after that to look at the details. And one of the things that came up as a surprise is we looked at the Ashkenazi Jewish trio, just the controls, regular genome controls. And you can see here, this is basically the different trios. You can see the Chinese trio in blue, Ashkenazi trio, and then a control, the Utah control. But you can see the ancestry specificity of some of these patterns basically shows the canonical and non-canonical motifs across the 5 to 8 kb of a normal telomere. And you can see it from different chromosomes. You can look at the clustering at the ends of the chromosomes of how they look. And so it's kind of really fascinating to see these telomeres have their own stories to tell, all these different variations that are present as well. And if you pull them out, you can actually get haplotypes to diplotypes. So you can look at what does each arm of each chromosome look like, haplotype 1, haplotype 2, and look at the distances between them and calculate them here. So it's actually really fascinating to see that, you think each arm of each chromosome might be relatively similar, but they actually have their own different haplotypes, which is kind of cool to see. these differences that are very subtle, but they're in the data. And so and that's actually at this point, this is Nanopore data that's 3 or 4 years old. These days, it's much cleaner, it's much more accurate, so it's much better, I'd say. But overall, good news we say about Twin Cities. Most things recovered, and we've since used these methods for a range of other missions. But I want to go back to an earlier question of, okay, someone tells you you have a really bad cancer to an extremely bad cancer that's not that satisfying. If we see bad methylation or dysregulated epigenetic states, couldn't we just modify them? Indeed, you can. So actually, there was a paper by looking at histone acetyltransferases by Charlie Gersbach in 2015 and 16 that showed you could bring the Cas9 system for CRISPR and then deactivate this Cas9 and bring something like a TET1 enzyme or a DNA methylation or histone acetylation, modify the different loci that you want to actually have this effect on. And this is something that was even a program by DARPA, which is this idea of like, what if we turn on protective genes before soldiers go into a radiological or chemistry danger zone, or maybe they have any sort of danger to their genome? Could you have a radio protection elements activated before they get irradiated? And this concept is something we've been following up on in the lab. And indeed, you can do CRISPR activation or inhibition. You can have these different constructs that will go in and land and turn on a specific locus or turn off a locus. And in this case, you can even have epigenome edited cells that can decrease the amount of cell death from half the cells dying to 27% dying with this radio resistance target. So it can basically turned on specific targeted genes that can confer more radio protection, showing that this works. So I think this is and this is a really good, I'll go back to this paper, very good review paper of the methods. And this paper just came out a couple years ago that I highly recommend it. So you can think about no longer being subjugated to one specific methylation change or histone change. You can, in theory, go back in and reprogram those specific sites, which I think is a fascinating time because I used to fantasize about this as a graduate student. Like, could someday you turn on or off genes in specific cells? The answer extraordinarily is yes, and you can do it today, actually. And so that seems abstract at first, but it's worth noting that this idea of turning on and off different genes over the course of time is something that already happens naturally in our body. This is, for example, fetal hemoglobin you can see in brown. You don't remember being a fetus. You probably don't remember being a fetus or an embryo, but if you remember either of them, please come talk to me. We should study you. But if you you don't recall, but don't need to recall, but your fetal hemoglobin was very active when you were first forming as an embryo, but was later turned off before you were born, and then became alpha and beta hemoglobin. And what's interesting about this is with people with beta thalassemia or sickle cell disease have problems with their hemoglobin. So if there's a problem, why not just bring back the gamma hemoglobin or fetal hemoglobin? And indeed, that's what's been done as a trial with the New England Journal of Medicine in 2021, showing that in blue, you can use the Cas9 system, actually in this case, you knock off the BCL11 repressor and then turn back on fetal hemoglobin. And this was done here, you see in blue. And it's actually now within 2 years, almost just 2.5 years later, is now an FDA-approved gene therapy for this CRISPR system to do somatic CRISPR therapies, which basically, even though it's a CRISPR deletion, you could call it a gene therapy. If you go back to this, it's really a regulatory therapy. You're turning off something that's a repressive element to then reactivate the gene, which is really a gene regulation question. And so this concept is really extraordinary to see that is now happening and is now FDA approved, which is extraordinary. And there's other places to use it. I talk about the same concept in my book, which I would totally shamelessly plug right now. But I want to close in the last part of the lecture a little bit about, these are genetic therapies, epigenetic therapies, but also what you're going to be doing a bit for the homework is also looking at RNA modifications as well. And just as there are dozens of DNA modifications, there are actually many, many dozens, even hundreds now known RNA modifications. You can see here 6-methyladenosine. You can see other variations here of these small chemical tweaks to RNA, just like we saw for DNA, that give you an ability to control when and where RNA is processed, stored, degraded. It gives you another regulatory layer on top of the RNA, just like DNA methylation gives you that regulatory layer on DNA. So it's really a great toolbox, essentially, for the cells. Same concept, though. Can you have a methyl group or a slight tweak and confer a change in function, stability, localization, or other features of a molecule? Indeed, the answer is yes. In particular, one of the first looks at this question we did with Sami Jaffrey back in 2012, which was to see, okay, well, if there's methyl 6-adenosine floating around in cells, is it across all tissues? The answer was yes. Is it developmentally regulated? It seems like more in the adult than early pups for mice. You can see here some of these are immunoblots of an antibody from M6A. And then we developed a method called MeRIP-seq, and also make it so it's kind of like ChIP-seq, but you do an enrichment with an antibody and pull down the RNAs that carry this methylation, elute them, and then sequence them. And when you call peaks, you can kind of find this little peak of RNA here and look at these spikes where that shows where M6A is across the transcripts. And so this worked pretty well. And then also, this is one of my favorite plots I ever made, the very first version of this plot, which was I looked across all genes and said, okay, is it enriched at the end of the coding segment, the UTRs? Where do we see this across all genes? And there's this really extraordinary enrichment at the end of the last coding sequence that we saw the spike. And this showed up again in human and in mouse. We can see this consistent feature of mouse brain and HEK cells. That was really mysterious because we thought, well, why is it doing this? What's happening? I'd never seen an enrichment plot quite like this, what's now called a metagene plot. And so at the time, we thought, this is, let's start to map these RNA modifications, figure out what they're doing. Ambitiously, we call it the birth of the epitranscriptome, which indeed what it was very much. And what we see now is it has a similar feature to DNA methylation and DNA modifications, where there are enzymes that confer and create these methylation, probably processing to, basically take away the methylation and process essentially the changes between RNA modifications, depending on their function and sort of cycling, and we think also depending on splicing. So it seems to be these factors that change these RNA methylations in this whole landscape. And today, actually, there's now over 400 RNA modifications that have been characterized. Some of them actually a lot of them are in tRNAs, controlling a lot with protein translation. Some of there are M5C, methyl 5-cytosine, M1A, M6A, different variations. There's the 7-methylguanosine cap, which most of you probably remember from your biology, but this is, of course, an RNA modification. Some of the RNA modifications were used for the COVID vaccines, which have to have a lot of pseudouridine for them to function. But there is an abundance of RNA modifications in almost every cellular system on Earth, and we've just really begun to map them out. So that's something that's been very evident really for decades, but become much more clear in the past few years. If you look at ModBase now, which is a really cool database that shows all RNA modifications, you can see it's now over to 421 RNA modified residues. So you can see by kingdom, by RNA type, by like SNO RNAs, tRNAs, non-coding RNAs, pi RNAs, many kinds of RNAs have these modifications. And one hard thing though is, okay, so we're all very excited. There's all these modifications. We published a critique. We downloaded actually this Alexa, a very talented student who was in the lab. She was really annoyed at the noise of MeRIP-seq data and said, I think people should have more than 2 or 3 replicates. And I was like, well, how many replicates do you think we need to get really consistent measurement of RNA methylation? And from our paper, it was really, you should probably have 8 or really 9 replicates before you start to get reproducibly detection of those peaks, which was kind of a terrifying plot to make because the vast majority of papers in the field had used 2 or best 3 replicates. Only rarely had they gone past that. So it was really a sad plot to make, but really a bit of an indictment of the field of epitranscriptomics, which we helped create. But you should always take a moment to take a harsh look at your own data and say, well, are we doing this reliably? And are there better ways? And what's interesting is MeRIP-seq was a workhorse for a good decade in the lab, but I couldn't be more elated to finally have the ability to look at all of the modifications and to be able to detect them all. So in particular, you can still do bisulfite treatment of RNA, which is called BizSeq. You can also do a G protective process, what's called GlorySeq, and then do A emanation, then D protection, and then sequence again. This is one method. Another method that's very accurate for detecting M6A. And also PRAISE, which is the same bisulfite and sulfite sequence. Then you desulfonate, you do reverse transcription, and you can see the sites that have pseudouridine here. But in theory, this is what we've been spending a lot of time on in the lab, and what I think you'll enjoy in the course is that wouldn't it just be nice if we could sequence a molecule and find all the modifications and set them in phase and know what's on the same molecule? Yes, it would be great. So there's all these methods, some of our own methods we've done work on for decades at this point. But this is something we've been, really thinking a lot for a long time, ever since I saw Spike talk about it about 10 years ago. And now, there's been some people who've done call to arms and say we should do this for every RNA everywhere. Of course, I agree with this statement. And now we can do these long reads. So the Promethean, as you saw before, looks like this. And really, just over the past 4 or 5 years, we'd say it reached maturity. We're getting from 86% accuracy, 89, 95. And actually, you'll see data in the homework and a bit very soon that we're now getting very high accurate reads and getting base modifications and you'll learn about all of them in your homework in the course. And this is something that we've been really, I think, excited about and looking at as a way to better understand the RNA biology, because now we can actually measure and see the biology with these methods. It's a very exciting time. And we can get long transcripts. So think about a lot of old sequencing methods were a few hundred bases. Now we can see 1, 2, 3, 4, 5 KB, sometimes even longer, and getting really, almost getting full-length transcripts in for a lot of samples we've looked at. And that's the backdrop of what you will start to see for the homework. One comment is it's very exciting, and it gives you all this cut capability at scale, but this is actually it's still not cheap. So we've been looking at various projects. It would still take hundreds of thousands of dollars or millions of dollars if you want to do this at scale. But we'll probably have an update by the end of the course of ways to do multiplexing of this prep, which will hopefully make things a bit cheaper, but it is not necessarily cheap per sample. But if you have the cash for it, it's great. And with that, I want to leave a few time for questions, but that is a bit of RNA and DNA modifications, and happy to take any questions.